This is a video in which we'll look at the chemical factors which affect the activity of enzymes. So here are the learning outcomes for this video. So first of all, we'll just understand the nature of the catalysis which enzymes can cause in terms of interactions between, well, two key terms here, a substrate and an active site. And we'll use this to explain the specificity of enzymes and then look at some data for how the rate of enzyme catalyzed reactions depend on substrate concentrations and make certain we can explain the shape of the graph you get in terms of the rate determining step within the mechanism. So relating it to ideas familiar to you from the rates of reaction topic. Okay, so just a quick reminder then of, in general, how catalysis works in chemistry. So a catalyst speeds up the rate of a chemical reaction by providing a new mechanism with a lower activation energy. Now that new mechanism, as you can see here, may well consist of several, in this case 4, separate steps. The total amount of energy needed to overcome the energy barriers for those steps is a lot less than the activation energy with the uncatalyzed reaction. As a result, many more of the collisions between the reacting substances have sufficient kinetic energy to be able to react, and the reaction therefore happens faster. Okay, so there's some key terminology we need to get to grips with. The first is that all enzymes possess a particular cleft, or if you like, hole within the enzyme structure known as the active site, which has a particular three-dimensional shape. Three-dimensional shape which enables a reacting molecule called a substrate molecule, or molecules, to fit in precisely. And so here's a very important word. We talk about the active site of the enzyme and the substrate as having complementary shapes. You can sort of see what that means here. It means that when you put one next to the other, they fit precisely together. They complete, in other words, a much larger shape. So what might that look like in a real situation? Well, here's a computer-generated model of an enzyme, and you can see here this cleft which represents the active site, and here's a substrate fitting into it. Now the important thing to realise is that not only do the atoms fit in perfectly, but also they bond at particular points to the surface of the enzyme, and that adds a kind of extra level of complementarity to the active site and the substrate. What we've got here, because of the bonding of the substrate and the enzyme, this is known as an enzyme-substrate complex, and you'll see that referred to when we look at the mechanism of enzyme-catalyzed reactions. So, if we can just focus in a little bit on what the enzyme-substrate complex will look like on a chemical scale, what we've got here is a chain of amino acids, and here are the side chains, and that chain is folded in a particular way within the tertiary structure, creating this cleft or active site. And you'll notice that amino acid side chains line the active site or the binding site, so that when a substrate molecule comes along, it fits into the space, but crucially, various atoms on that substrate molecule are able to bond via, for example, hydrogen bonding or ionic bonds to the amino acid side chains that line the active site. And so both the bonding and the three-dimensional shape are complementary. So it's important to realise that that bonding will depend not just on the shape of the active site, but also on the exact position of these amino acid side chains. Okay, so then we have to understand the first of these key terms, specificity. And this is simply saying that only one particular substrate, one specific substrate if you like, is able to fit into and bind to that enzyme, and hence there's only one substrate which has a reaction catalyzed by any particular enzyme. Other substrates with different shapes, different structures will be unable to fit and bond, and hence they will not undergo a reaction catalysed by the enzyme. So this is the key statement here that you'll need to put into any exam answer. And you need to realise that it's not just the fitting in, but it's also the binding of the substrate to the enzyme. Now that specificity can be at quite a sophisticated level. Clearly in a diagram like this you can see that that substrate there is able to fit into the enzyme, but the enzyme is able to actually distinguish between, for example, optical isomers. So here are two molecules that have identical structures except that one is a mirror image of another, but because there are particular active sites at these binding sites within the enzyme, it's only one of the optical isomers which actually has atoms in the correct orientation to bond to the amino acid side chains in the binding site, and as a result only this optical isomer is likely to be able to react as a result of the enzyme being exposed to it. And that's quite an important point. Okay, so we talked about the mechanism of enzyme catalysis. So we've seen the idea of the enzyme and substrate reacting to produce an enzyme substrate complex. That's actually a reversible process, interestingly enough. However, once that is formed, we can then get a reaction to form a product, and finally the products leave. So we've got binding, reaction, and then the products unbinding and leaving. Similar in some ways to the mechanism that you might remember for heterogeneous catalysis back in the AS year. Now we can represent that reaction symbolically. Obviously we wouldn't dream of trying to write chemical formulas for enzymes, but we can represent enzyme by E, substrate by S, and so here's the first step, the formation of the enzyme substrate complex. Here's the second step, the reaction of the substrate within the enzyme to form a product, and here's the third step, the products leaving from the enzyme. Now clearly there are two substances involved in the reaction, two reactants, the enzyme and the substrate, and so the rate of reaction could in theory depend on either or both of those concentration terms. We'll have a look in the next section of the video in terms of what the pattern is for the way in which rate of reaction depends on those concentration terms. So here is a graph that really you'd be well advised to memorize the shape of. It shows how enzyme activity depends on substrate concentration. Now this is slightly different to the graphs that you might expect in a simple rate of reaction investigation of rate against concentration of a reactant. It's worth, I think, splitting the graph down into two sections. Let's look at the section first of all where the substrate concentration is low, and as you can see the rate of reaction increases as substrate concentration increases. We get a nice straight line graph. So can you stop the video and think to yourself what that tells you about the order of reaction with respect to substrate concentration? So hopefully you were right, and you said that at low substrate concentration the order is first order because we get a proportional relationship between rate and substrate concentration. So can we therefore write a rate equation for this reaction? Well, yes we can, if we use the fact that the reaction is always first order with respect to enzyme concentration. So putting those facts together, can you write a rate equation to show how rate depends on enzyme concentration and substrate concentration? Pause the video and do that now. And so we should end up with a rate equation looking like this. First order with respect to both enzyme and substrate concentration. Okay, what's happening in this section of the graph here? Well, after it passes through a particular point of threshold substrate concentration, suddenly we're finding we get a horizontal line. So you might like to pause the video while you think about what order that suggests about substrate concentration in this region of the graph. And what you should have realised is that it's zeroth order here. Why? Because as you increase rate, as you increase concentration, there's no effect at all on the rate of reaction or activity of the enzyme. And so now can you write a rate equation to describe how rate depends on enzyme and substrate concentration in this region of the graph. And so yes, now the rate depends purely on enzyme concentration. So we may need to be able to explain that graph. So key thing to realise is that we've got these 3 steps in the reaction, and those 3 steps have very different rates. And the key thing to remember is that the rate determining step is actually the second step, the reaction of the enzyme substrate complex to produce the enzyme product complex. That's the one that happens in the actual enzyme itself. So the point will be, therefore, that the rate of that reaction, and hence of the overall enzyme reaction, is going to depend on the rate of on the concentration rather of enzyme substrate complexes. And we then have to consider, well, what factors affect that concentration? Well, because both enzyme and substrate occur before the rate determining step, you imagine that those are going to both affect the enzyme substrate complex concentration and hence will affect the rate of reaction. And so the expected rate equation would be the one that we see below, indeed the one that we wrote for low substrate concentration in the previous part of the video. So that fits in with what we know about mechanisms that reactions that occur before the rate determining step or in the rate determining step will be first order. However, we've got a different situation to conventional reaction kinetics here, and that's illustrated by this kind of diagram here. What we've got is looking at the situation in which you have 2 situations, one in which the enzyme is present in excess, where there's very low substrate concentrations, and then a more extreme situation here where there's such a high substrate concentration that it is present in large excess. Now, in the low substrate concentration situation, most of the active sites are unoccupied. So here's an enzyme substrate complex here, and you can imagine that the concentration of that is going to purely depend on how frequently substrates and enzyme collide. So unsurprisingly, the concentration of the enzyme substrate complex depends on both enzyme and substrate, and therefore the rate will depend on both enzyme and substrate, as you can see there. So now let's consider this situation here. So now, if you noticed, all of the active sites are occupied. So here are the enzyme substrate complexes, and now any further collisions between substrate molecules and active sites, substrate molecules and enzymes, is not going to increase the concentration of the enzyme substrate complex. It can't do, because there's no more enzymes to form those complexes. So as a result, the only thing that can affect the enzyme substrate complex concentration is the concentration of the enzyme. And so as a result, the rate ends up depending purely on enzyme concentration. It's quite a complex idea, but the key to it is all to do with looking at whether or not active sites are mostly unoccupied or mostly occupied. If you can get across those ideas, then in most cases you will end up getting full marks in explaining the shape of the curve. So that concludes the coverage of how concentration of substrate affects enzyme activity. In a second video, we'll look at some other factors that affect enzyme activity as well.